Welcome to OsteoBytes. Thank you so much for joining us today. We'll give a couple seconds for everyone to get settled into our virtual room. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBytes, we are joined by Dr. Chris Richards, one of our 2024 Outsmarting Osteosarcoma grant recipients. And he will provide an overview of his funded work on macrophage engineered vesicles to treat pediatric osteosarcoma. Thank you so much, Dr. Richards, for joining us on OsteoBytes today. We are thrilled to have you. And we're also so happy to have Walker back as our panelist. I'm feeling some major UK Wildcat vibes today. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom, and then we will share those with Dr. Richards when he is done with his presentation. A little bit more about our guest today. Dr. Richards is a professor in the Department of Chemistry at the University of Kentucky, where he also serves as the director of the Light Microscopy Facility and the director of the Bioelectronics and Nanomedicine Center. His lab has developed novel nanoscale materials for therapeutic delivery and in vivo sensing within the central nervous system. Incorporating these materials with nanofabricated devices has enabled his lab to develop new platforms for interrogating biological systems in cell culture, isolated tissue, and in vivo. Research in the Richards Lab also focuses on the development of ensemble and single molecule fluorescence spectroscopy techniques for the study of complex biological systems. His lab has recently applied this approach to study substance use disorders, along with the development of therapeutic delivery platforms for cancer and spinal cord injury. Dr. Richards received his bachelor's degree in chemistry from the University of Maine and his PhD in physical chemistry from the Georgia Institute of Technology. And he was also a postdoctoral researcher at Caltech, where he was also a Beckman postdoctoral scholar. Thank you so much for joining us today, Dr. Richards. We are very much looking forward to your talk. Just a few announcements before we get started. Save the date, Factor 2025, June 26th to 28th at the Grand America Hotel in Salt Lake City. Registration will open in early February, but abstract submission is now open, and the submission deadline is January 10th. So hope you all consider applying and submitting a abstract for that. And if you are looking for the perfect holiday gift for your osteo warrior or even possibly a colleague, we've got you covered. Even though registration doesn't open till next year, you can purchase a gift certificate for Factor 2025 now. Some of the best gifts are experiences, not things. And this is a very rich experience that will be a gift that keeps on giving with all the connections that are formed at the conference. So I'll put some more info in the chat about that. And also in our MIB Holiday Boutique, you can order MIB holiday cards that showcase artwork from 6 talented osteo warriors who have created original artwork for our holiday card collection this year. Each box has 12 cards. They're perfect for sharing joy this season, and I'll put some more information in the chat about that. Walker, could you go ahead and introduce yourself, please?
Yeah, hi, my name's Walker. I'm part of this year's Junior Advisory Board and have been for the last few years. I am actually a current University of Kentucky student, so I'm really excited to hear more about today's presentation. I've been tortured by the chemistry department for about the last 3.5 years, so I'm very, very excited to see all the good things that are going on there. And so you can take it from there, Dr. Richards.
Okay, thank you very much, Walker. Let me see. Okay, so I think you'll see for the next couple of slides that I am not what you would think of as a traditional cancer therapy researcher. So I want to point out Jill Kolesar on the upper left-hand side, who used to be at the University of Kentucky till about 4 months ago, then abandoned us and decided to become the dean of pharmacy at the University of Iowa. But all of the cancer work that we do in my lab really started with Jill about 5 years ago. And we have an ongoing collaboration, even though she has left us here. That collaboration is ongoing. I guess I should have started this video already, but let's start this. So what my lab focuses on is the development of nanoscale vesicles that we engineer, typically from cellular membranes. And this movie basically just shows a time lapse of macrophages, which are in green, interacting with vesicles, and you'll see these small membrane vesicles arriving, interacting with the cell, getting taken up by the cell, and then slowly merged into the membrane over time. But this really is a visualization of everything that we're looking to do with our platform. There we go, I moved. So my lab traditionally has been more of a neuroscience lab and a somewhat of a hardcore photophysical chemistry lab. We develop a lot of in vivo techniques to do spectroscopy, allowing us to visualize biological processes, a lot of single molecule methods, substance use disorder. But starting about, I want to say in 2018-ish, we started working with Jill for what you see in the lower left-hand corner, which is our targeted delivery systems. Since that time, I would say it's probably become about half of what my lab does. We really have gone in that direction, in part because of some of the promising results that we've seen that have come out of that collaboration. So when we think of vesicles, in my lab, we think of them a little differently than what most researchers think. I think you can put vesicles on two ends of a spectrum. On the one end, you have synthetic vesicles, like lipid nanoparticles, liposomes. Those of you familiar with those types of treatments know that there are a number of cancer therapeutics that are based on liposomes. For example, there's a popular one where they're loading liposomes with doxorubicin, for example. But the lipid nanoparticles, for example, have also come back into favor with the development of some of these mRNA-based vaccines as delivery systems. So they're on the one side where they're completely synthetic. Unfortunately, they have typically a high level of immunogenicity because they're not natural to the body. And on the other end of the spectrum, you can think more about endogenous vesicles, such as exosomes or extracellular vesicles, which have really garnered a lot of attention over the last 10 years, because there are a number of people that are very interested in using this type of technology of a cell producing a vesicle that can then be harnessed, loaded with a therapeutic or functionalized, and then used to target back to the cancer. And the reason for that is these types of vesicles, these endogenous vesicles, are used for cell-to-cell communication. So there's this idea that they preferentially go back to the cell of origin. So you can imagine if a cancer cell produces a vesicle, it may actually go back to the cancer cell. There are some issues with these, which is where we come in. They are messy and complex mixtures of vesicles. So if you're trying to extract them from a patient, for example, you're going to get vesicles from a wide variety of cells with a wide variety of properties and even a wide variety of sizes. And if you want to produce them in a lab, you need a lot of cell culture, and they're produced at very low yields. So we sit somewhere in the middle of these, where we actually generate vesicles from cellular membranes that mimic the properties of these exosomes. And the reason we want to mimic the properties of these types of vesicles is because they have shown promise for cancer therapeutics. And one of the things that exosomes have been shown to do is to be able to modulate the immune system, right? So they're immunomodulatory, and depending on the type of cell that you get the extracellular vesicle or exosome from, you can push the immune system or immune cells towards either a more pro-inflammatory or a more anti-inflammatory phenotype. And some types of cancer exploit this, where cancer cell exosomes can actually interact with the immune system and inhibit the immune response. However, if you're looking at exosomes from certain type of immune cells, that can actually be immunostimulatory at the same time. So the question that we started was, well, can we generate vesicles that mimic the properties of exosomes that can be produced at very high yields, that'll maintain the properties of exosomes in terms of targeting specificity, have the low immunogenicity, that are inherently biocompatible, and that can also modulate the immune system. So that was sort of our jumping off point to develop the platform that we utilize. And a little bit more on the motivation of that is there was some work with exosomes where the exosomes derived from M1 macrophages, so what's commonly a pro-inflammatory macrophage, showed a lot of promise in anti-cancer treatment, where they were able to initiate a pro-inflammatory immune response that would basically prime the system for some of these other immune therapies like immune checkpoint inhibitors. So the way we stumbled across this is that my lab had developed this technique where we would make vesicles from the brains, mostly of our mouse models, or cardiac tissue. And the reason we were doing that is because we like to do single molecule spectroscopy, which means we find an individual protein, we isolate it, and then we shine a laser on it, and then we try to get some properties like the assembly of the membrane receptor, which is important for some membrane receptors in the brain because depending on how they assemble, then their properties change dramatically. So we developed this technique where we would take the brain out of a mouse, and then we would rapidly, for lack of a better word, blow it up and generate these small vesicles that would trap the single proteins that we were interested in. So I was sitting in a talk at the pharmacy department at UK, and there was someone that was giving a talk super excited about exosomes. And the more we talked about exosomes and how he was going to basically cure cancer and save the world with exosomes, it dawned on me that the vesicles that we make via our single molecule technique were very, very similar to what an exosome was. So that basically was where we started. The same day, I went and banged on Jill's door and said, hey, what about this idea of using these vesicles that we have as kind of a fake or an exosome mimic? And let's see if we can generate some of the same properties that an exosome would, but we can do it in a way where we can generate these cells, engineer them to have properties a little more easily than you could in an exosome, but also generate them maybe at 100 or 1,000 times the yield that you could with an exosome, because they become a feasible therapy. So we make vesicles a lot of ways. I will talk about the ones on the left, which are our least favorite, then the one on the right, which is our favorite. One of the ways that we can make vesicles is we just take cells and we push them through a membrane with tiny holes. So you can see these holes up here. Let's say they're about 200 nanometers. Our cell is, say, 25 microns. And then we'll just put a lot of pressure and we'll push it through these holes, and it will tear the membrane into pieces. And what happens is you end up with a little flat membrane segment, but you have around the edge these hydrophobic regions that do not like to sit in an aqueous solution. So what they do is they fold back up on themselves, and then they form our vesicle. So we can do it by pushing them through a membrane, very analogous to how you would do extrusion for a liposome. We can do this via sonication, where we basically just sonicate a solution of cells until they break apart. There are some issues with these techniques, even with down homogenization, which we've used, is the throughput's usually a little lower. It's hard to scale these. And then all of these processes are mechanical processes. So we're generating a lot of heat. And our concern is that sometimes this is going to damage the proteins on the outside of the vesicle. And we'll see later on that these proteins on the outside of the vesicle are very important for the properties of the vesicle. So our favorite technique right now is to use nitrogen cavitation. What that means is we can take a vessel as big as a liter. We can put billions of cells into this vessel. We pump in very high pressure nitrogen. Then we let it sit for a little bit, and then we rapidly release that pressure. And when we release that pressure, the nitrogen bubbles tear the cells apart. And what you end up with is a whole bunch of cellular debris, but a whole bunch of vesicles as well. Then we can go through a process of centrifugation, and we end up with vesicles at the end that are very reminiscent of what a typical exosome or extracellular vesicle would be. So what I'm going to talk about and what we're funded to do through this grant is to actually generate vesicles from macrophages. We're going to call them macrophage engineered vesicles, or MEVs, as we have up here. we started with a bunch of names. I think we started with cabosomes was our favorite at first. Horrible name. I blame Jill for that. Jill has come up with all the names. But we abandoned that. I think we even got criticized for the name in one of our proposals we sent to the NIH. They thought it was a goofy name. But we settled on macrophage engineered vesicles. And the whole idea is if we have a tumor microenvironment which is supportive of the cancer cells, basically we have an abundance of tumor-associated macrophages, which of course have more of an anti-inflammatory phenotype, and they are not generating conditions to attack the cancer. What we want to do is we want to sensitize this environment by delivering these macrophage engineered vesicles that can modulate the polarization of our TAMs, basically by moving them from an anti-inflammatory phenotype to a pro-inflammatory phenotype, making them anti-cancer. So the first question we wanted to ask is, can we flip suppressive macrophages into an anti-tumor macrophage? My students have discovered BioRender, by the way, so they love to make cartoons. So I include a lot of cartoons in my talks now. If you don't use BioRender, then I encourage you. It's relatively cheap, and they make some remarkable cartoons sometimes. But the idea for most of our studies in mouse models, we're generating bone marrow derived macrophages, going through that whole nitrogen cavitation process, and then going through our serial centrifugation. And what we end up with is our purified MEVs. We can do this either from M1 vesicles. We'll call them M1, even though that might be slightly out of vogue at this point. Pro-inflammatory macrophages, we'll label those as M1 EVs. And for the ones from M2, we will label as M2 EVs, EV standing for engineered vesicle, not extracellular vesicle at this point. So one of the things we did early on was to look at, would our vesicles have the same specificity for delivery that has been demonstrated by exosomes? I guess I'll tell a short story since this is more of a casual presentation. We started off using cancer cells as our source of vesicles because there's a lot of literature, some that I pointed to previously, where exosomes and extracellular vesicles from cancer cells are being used as a therapeutic. So we took the same strategy where we were generating vesicles, our engineered vesicles from cancer cells. We also went to the NIH with that, and they hated it. They hated the idea of delivering something from a cancer cell back potentially to a patient, where there was the potential to basically increase your metastatic potential, let's say. So that idea got shot down pretty quickly, and then we ended up transitioning to the immune cells. But here, this is some stuff from when we first started. So the idea was, if we take a cell and we deliver vesicles here in red to the purple cell that are from a different cell type, how is the uptake, or how is the specificity of the delivery versus vesicles that are in purple delivered to the same cell they derive from? And what we can see, if we deliver just a couple of common cell lines, HEK cells versus A549 cells, HEK cells go back to HEK cells quite well. If we look at a macrophage cell line, raw cells, versus a cancer cell line, also goes back to, they go back to the macrophage cell line. That's also shown down here, where you can see we get a lot of delivery where we've labeled fluorescently our vesicles. So anything that's in red is a cell that is fluorescent, that has had a vesicle delivered to it. And then over here is what we see for the A549 cells. And then on the far right, the graph shows delivery to our macrophages versus delivery to a cancer cell line. And again, making vesicles from the cancer cell line, they are preferentially trafficked back to the cell of origin, very similarly to what has been shown in the literature for exosomes. So the next question we wanted to ask with our macrophage engineered vesicles is, can we modulate the polarization of macrophages? So what we do is we start with a culture of M2 macrophages, so macrophages that are in an anti-inflammatory phenotype. I'm having trouble seeing row 2. I don't know if you'll be able to see row 2, but here INOS is a marker for a pro-inflammatory macrophage, and you can see you have very little by the absence of green. This should be purple. I can just barely see it on my screen, maybe because it's in display mode. But you can see that they're M2 macrophages, and these are just the nuclei. And then what we're doing is we're adding more and more vesicles. And as we increase the number of vesicles, what you can see is that INOS expression increases significantly, indicating that we're moving from an M2 to an M1 or an anti-inflammatory to a pro-inflammatory phenotype for these macrophages. And that's quantified on the right-hand side here, where you can see as you increase the number of vesicles, we increase our expression of INOS or our pro-inflammatory behavior. And then we get a reduction in our anti-inflammatory marker. Pro-inflammatory cytokines, pro-inflammatory macrophages basically give off pro-inflammatory cytokines. So we also measured cytokine release. TNF-alpha is a common pro-inflammatory cytokine that corresponds to a pro-inflammatory macrophage. And very similarly, as we increase the number of vesicles, which were derived from our M1 macrophages originally, we can flip an anti-inflammatory towards a pro-inflammatory behavior. And then a Western blot down here that basically shows the exact same thing. I sometimes tend to get over on time, so I'm just double checking. Okay, so we wanted to then look at more of a panel of pro-inflammatory cytokines. So this is a little messy, but we'll walk through it real quick. If we look at cytokine release by a pro-inflammatory macrophage in culture, or we'll say a culture of pro-inflammatory macrophages, we get the release of all kinds of pro-inflammatory cytokines, and then we can also look at NO down here. So this is kind of our baseline of what a pro-inflammatory macrophage is in culture if we hit it as hard as we can with LPS, for example. And then if we look at what an unpolarized resting macrophage looks like, you can see we produce pretty much no pro-inflammatory cytokines. If we shift our resting macrophage to an anti-inflammatory macrophage, or it's classically called an M2, then you can see we also get very little to no production of pro-inflammatory cytokines. If we take our vesicles that we derived from M1 macrophages, so these engineered vesicles through the nitrogen cavitation process, and we deliver them to our M0s, you can see we do get the uptick of some pro-inflammatory cytokines, showing that along that continuum from anti-inflammatory to pro-inflammatory, we are pushing them a little along the way. Interestingly, we get a much better response from our anti-inflammatory macrophages. So if we have an anti-inflammatory macrophage, much like a tumor-associated macrophage, and we deliver our vesicles to it, what we see is we have pushed it much more towards a pro-inflammatory phenotype, because it's now producing a number of these pro-inflammatory cytokines, like TNF-alpha, for example. And we can actually go the reverse direction as well. And if we, again, have an M1 macrophage culture producing these pro-inflammatory cytokines, and we make vesicles from M2 macrophages, we can give them, and you can see that we knock down the production of a lot of these pro-inflammatory macrophages, like TNF-alpha, for example, goes from our normal level down to virtually nothing. So it shows that we have this bidirectional ability to shift macrophage phenotype either towards pro-inflammatory, which is the goal when we're talking about a cancer treatment, or back the other way towards an anti-inflammatory, which we have other applications for that. So over here, we've done the same thing with microglia and astrocytes. It works roughly the same way. In fact, it works a little better on microglia when we're delivering macrophage engineered vesicles than it even does on macrophages. So one of the questions that one of my students really got interested in was what's driving this repolarization. That might be because he went to a conference and somebody beat him up a little bit, asking him why are these vesicles working to flip macrophage phenotype. So he was doing a number of experiments in the lab. One of the things he was doing is he was separating out vesicles made from the endoplasmic reticulum versus those made from the plasma membrane. So the ER endoplasmic reticulum is an intracellular organelle. But when we generate our vesicles, we typically have a mix unless we separate them. Some are from the cell surface, and some are from the ER, and some are from other intracellular organelles. But what he noticed is when he flipped or when he split these into ER and PM specific, that the ones from the endoplasmic reticulum were actually much better at flipping macrophage phenotype than the ones on the plasma membrane. So what that led him to think about was that there was something specific about the surface of these vesicles that was likely responsible for driving these changes in our macrophage phenotype. So one of the first things he looked at was residual cytokines. One of our ideas was maybe we are just trapping pro-inflammatory cytokines inside of our engineered vesicles, and by doing that, we're then delivering them to these anti-inflammatory macrophages, and this was causing them to flip towards a pro-inflammatory phenotype. So he measured the amount of pro-inflammatory cytokines in solution. When we generate our vesicles, we have virtually nothing. And then he went through a process of freeze fracturing them, basically taking the vesicles, putting them in liquid nitrogen, rapidly warming them up and going back and forth. And what that does is it pops the vesicles open and it releases all of their contents into the extra vesicle solution. And then we also measured the concentration of IF gamma, for example, which we came out to about 40 picograms per milliliter. So then our next test was, okay, let's take 40 picograms per milliliter and see what that does to an anti-inflammatory macrophage. And our pro-inflammatory versus anti-inflammatory phenotype here is measured based on TNF-alpha. So you can see an M2 sits down here, and then if we add 40 picograms per milliliter of IFN gamma, we get very little transition away from our M2 towards our normal M1. And if we put that onto a scale with what our MEBs induce, you can see that our engineered vesicles push us much further towards a pro-inflammatory phenotype than what you could get even by adding something like 40,000 picograms per milliliter, much less 40 picograms per milliliter. So this led us to conclude that it's very unlikely that cytokines are what are producing this ability for our vesicles to modulate immune cell polarization. So the next culprit that we looked at was membrane proteins on the surface of these vesicles. So exosomes, there's a number of standard exosome markers, CD9, CD63, a lot of membrane proteins that have been associated with exosomes and extracellular vesicles. And when we look at our engineered vesicles, we find almost the exact same membrane proteins as we do when we look at an exosome, for example. So this is basically looking at a couple of markers like an ER marker, and a plasma membrane marker, then a cytosolic marker. All of these are shown in the cells themselves, but our engineered vesicles don't have the cytosolic marker. So our next thing we wanted to look at was, is it the surface proteins that are basically driving this repolarization capability? So of course, if we look at kind of our model here of our vesicle or an exosome, it's populated with a number of these membrane proteins that could interact with the cell. and when they interact with the cell, initiate some type of signaling cascade that, depending on that interaction, could drive things either towards a pro-inflammatory or an anti-inflammatory phenotype. And this has actually been implicated in the literature for how exosomes mediate macrophage polarization. So it's not a surprise that we kind of went down that road as well. So I thought the student derived a pretty clever experiment to test this. What we start with is a vesicle that has a sealed membrane and then has a number of proteins with an extravesical region of the protein kind of sticking off the vesicle, then and transmembrane domain, not shown here, but we'll say a luminal vesicle domain as well. And what they did is they basically did a proteolytic digest where they used a protease to chop up all of the extracellular region of the protein. So what you'd be left with is fragmented protein, and then just the remnants of those proteins on the outside. And if we look at his Western blot here, we can see from Western of the M1s, we have a number of these proteins. And when we look at the MEVs, we have a number of these proteins. But once we do the proteolytic digest, we still have our vesicle, but we've knocked down all of these membrane proteins because they've basically been eaten up on the outside. So then he did his first experiment where he looked at delivery of our engineered vesicles versus our proteolytic digested engineered vesicles. And he was a little disappointed because they were still taken up by the macrophages. There was a very clear difference, but not a substantial difference, which may be not that much of a surprise because macrophages are great at engulfing stuff, right? That's what they do. But where we got really excited is when we started to look at the ability of these vesicles to flip macrophage phenotype. You can see that our regular engineered vesicles were able to take an M2 or anti-inflammatory macrophage and flip it towards an pro-inflammatory macrophage, as demonstrated by the ability of those flip macrophages to produce TNF-alpha. However, when we treated them even with increasing amounts of our digested vesicles, we've completely eliminated that ability to flip macrophage phenotype. So what that told us is that we had basically, we're generating vesicles, and the properties of those vesicles in terms of being able to flip macrophage phenotype was really driven by these membrane proteins that were expressed on the surface of the vesicle. This isn't necessarily the pathway that's important, though it may be. This, I think, was the best guess of the student at the time. But the idea is still the same, whether it's this particular interaction or not. But some protein on the surface of our vesicle interacts with a protein or proteins on the surface of our macrophage, initiating a signaling cascade that then leads to a change in the polarization of the macrophage, whether it's towards anti-inflammatory or pro-inflammatory. We were very, very interested in figuring out which of those membrane proteins were responsible, because our idea is that we want to program those into the surface of our vesicle, and we can do that in a much easier way than we can with an exosome. But the idea was that maybe we have 100 copies or 10 copies of this protein, and we'll make a vesicle with 1,000 copies of this protein. So now we have this super vesicle that can basically flip macrophage phenotype much more efficiently. Instead of having to deliver 10 to the 10 vesicles, maybe we can deliver 10 to the 8 vesicles. So from one treatment from a patient or one extraction of the cells, which I'll get to as well, from a patient, instead of being able to give them 2 weeks of treatment, maybe if we can supercharge these vesicles, now we can give them 6 months of treatment or a year of treatment just from one apheresis. So in terms of applications of these vesicles, one of their primary applications, of course, is osteosarcoma. And I will show you some data on that. We also are looking at and other applications. I'm going to bring up ovarian cancer in a minute because I think there's something that is very relevant to, and that is going to help push us forward with our osteosarcoma work. But we're basically making vesicles. We have some other projects with spinal cord injury, and one of our pushes and what this grant covers is really moving us from the laboratory to where we can do GMP manufacturing. And being able to get these into a clinic and then actually move towards a clinical trial. So what we see in terms of osteosarcoma is that we're looking at two treatments. One, which we're going to call empty MEVs or empty engineered vesicles. So there's no therapeutic inside. These are just vesicles with the idea that we can deliver them to the tumor microenvironment. And in doing so, we can flip macrophage phenotype and take a cold tumor and turn it into a hot tumor, if you like that terminology, and just through that process generate cancer cell death. And the other treatment we're looking at is a therapeutic loaded cisplatin or another therapeutic where we can simultaneously target the tumor microenvironment, flip macrophage phenotype, but also deliver a therapeutic to that region, which will kill cancer cells as well. So here's some osteosarcoma data. I do want to point out that I think all of this data here was collected by Namrata Anand, who was in the Kalesar group. So what we would do is we would generate vesicles. We used a model that was bioluminescent, so then we could basically have a readout of the bioluminescence for the tumor size. And then what we did is we treated with control, free cisplatin, empty vesicles, and cisplatin treated vesicles. And what you'll see is that over the course of the treatment, the vesicles, whether they were free or loaded with cisplatin, behaved or were equally successful as free cisplatin in knocking down the tumor growth. Where I think we got really excited is that the mice don't tolerate the cisplatin treatment very well, much like a human patient does not tolerate the cisplatin treatment very well. And what we saw is after a very short treatment, the mice started to lose weight, and eventually we had to sacrifice them. However, they tolerated the cisplatin loaded or the empty MEVs perfectly fine, and we saw no loss in weight and no need to sacrifice the animals in the same way we did with the free cisplatin. And this is just a percent survival curve showing that both the cisplatin and empty MEVs survived significantly longer than the cisplatin, mostly because of the animal's inability to withstand the cisplatin treatment. The other thing that's important for this was basically looking at metastatic nodules to the lung, and what we showed was that here's control, free cisplatin also knocked down a number of modules per mouse, but you'll see that the empty MEVs were equally as successful, more so than the cisplatin MEVs either, meaning the vesicles that were loaded with cisplatin. We've gone back and forth as to why the cisplatin was not as efficient or efficacious as the empty MEVs. And our best guess right now is that when we're targeting that tumor environment, I don't think I show much in the way of imaging beyond this one, but you'll see that our vesicles, which are labeled, are going towards the tumor preferentially. They also go to some of the other organs, but preferentially towards the tumor, and that's because they're targeting the macrophages. And what we think is happening is probably the cisplatin is killing all of our macrophages. So whatever benefit we get from our macrophages, we're now losing that because we're eliminating our macrophages with the delivery of the cisplatin. So my lab loves technology development. So there's everything is kind of focused around this idea of technology. And I think there's a number of exciting things that Jill and I have in the pipeline that directly will benefit our osteosarcoma work. Number one, we started a collaboration with Christian Cappuccini at the University of Wisconsin, and he has a nice syngeneic model of osteosarcoma, and we are going to basically start sending him vesicles where he's going to treat his model, and that will tell us, a mouse with a complete immune system, how effective our vesicles are. Here, this is a little bit off the wall, but I had a student who came from a lab where she did bacteria work, and she convinced me that she wanted to make vesicles from bacteria. She tried it in a different cancer model. I'll show that data in just a second, but I have another student who is super excited about taking that same work and is now working on the osteosarcoma project, and is actually going to deliver bacterial vesicles to our models of osteosarcoma, and see what kind of results we get, and see if we can replicate the success we've had with another model. And program vesicles, I talked about that a little bit. We have a couple of targets. We've spent a lot of time trying to figure out which membrane proteins are important. We have a couple that we think are important, but that's actually been not as straightforward as we expected it to be. We have a funded clinical trial that's about to start on ovarian cancer, and a lot of that grant is designed to get these vesicles ready for FDA approval. So we think that we're going to be able to piggyback the osteosarcoma work off from that, right? We're going to do all the hard work with this ovarian cancer model. And then once we come out of that clinical treatment, then we're ready with our vesicles all ready for FDA approval, and we can jump with any luck to an osteosarcoma model, as long as we can get something to run that. So the bacterial vesicle idea that the student had was basically we generate vesicles in the same nitrogen cavitation way from an E. coli that has limited LPS. And then we will mix them with a cancer vesicle, so we have a fused vesicle. Or alternatively, what we've been doing is we've been engineering our bacterial vesicles so they have a cancer antigen on the surface of the bacterial vesicle. So the idea is quite simple, right? The bacteria will elicit a very strong immune response. So if we can engineer something that's associated with the cancer into that surface, now we've directed the immune system to also target the cancer as part of the response to this bacterial invasion. And so we're taking advantage of this pathogen recognition, but because we're using vesicles, there's no ability to replicate the bacteria. So we're not worried about a bacterial infection. I think I'm closing in on time, so let me just jump through this. What's pretty exciting in terms of flipping macrophage phenotype, we can do it at orders of magnitude lower numbers of vesicles than we can with our MEVs. That's kind of shown here for each of these pro-inflammatory cytokines. Let me jump through. I'll just show this real quick. The student worked on a melanoma model because that was the one syngeneic model we had access to. So we need an intact immune system in the mouse in order for this to work. And what you can see, she had a number of different types of bacteria, some mixed with cancer antigens, some mixed with cancer membranes. But what you can see is this was a pretty aggressive model. After 21 days, this is where we were with our control, and this is what we had knocked down with our bacterial vesicles. So right now, as I said, the new student is up and running on this, and I think has animals that we are getting in collaboration. And we're going to start trying this on osteosarcoma probably within the next 2 months, I would guess. I've talked a bit about program MEVs. The whole idea is if we find something on the surface that's important for repolarizing, then we can just overexpress that on the vesicle. And at the same time, there are a number of surface modifications that we can make. And we've tried all of these, everything from folate to PAM3K, 3CSK4, in order to make our vesicles a little more advanced. This is some of the preliminary data from ICAM1, which is one of our program targets. We've completed making a cell line of these now. This was not with a cell line, but just overexpression. And you can see we can, if we take our M1 vesicles that overexpress ICAM1, we can, in comparison to MEVs that were not programmed, we can elicit a bigger pro-inflammatory response. And we're excited with the new cell line that we just generated, because now we have much higher levels of expression, and it's across all of our vesicles. So we're also probably within the next 2 months should know how those work in the osteosarcoma model. And one thing we're excited about, not only for this because of the ovarian cancer implications, but also, like I said, for what it's going to do for our work with the vesicles on osteosarcoma. Jill and I have received an award to commercialize MEVs for ovarian cancer through ARPA-H. So it will pay for a clinical trial and pay for us to basically get these vesicles ready for FDA approval and hopefully into a clinical trial for osteosarcoma. I think with that, I should thank Jill, since she is the person who has dragged me into the cancer therapeutic world, funding from MIB Agents, of course, along with some other sources. And this is my group trying to look cool on the stairs. And I'm happy to answer any questions.
Thank you so much. And definitely the cartoons were very helpful in your presentation. So glad they figured out a biorender. So everyone, please feel free to add any questions to the Q&A. I think we already have a couple. Walker, if you want to start with those.
Yeah, the first question that came in the Q&A asked, have you considered autoloading your MEVs with apoptosis-inducing agents that have been shown in brain cancer models to also be expressed on the surface of exosomes?
We haven't, but we're always interested. The dirty secret of what we do a little bit is we go out and we look at the exosome literature, right? So, and that's the first thing I tell a new student: go out and look at what people have done with exosomes, bring it back and do the exact same thing with our vesicles. We haven't considered that, but I've got a pen, and I'm sure there's a student that'll be excited to look at that literature and try it.
I had a question. So I know that it's really difficult to get immune cells into the interior of the tumor, or like specifically for osteosarcoma. And so I guess what I was curious about is when you get these vesicles to go in and repolarize the macrophages, are they able to penetrate to like the core of the tumor, or is it more of something that's happening on the outside and then the core of the tumor is still like cold, as you were talking about?
So with the MEVs, we believe they're penetrating. We don't have as much evidence as we do with the bacteria, because part of the problem is we haven't used the syngeneic model, right? We need a model where we have an intact immune system to really answer that question. So that's a big part of what we're doing with Christian. With the bacterial vesicles in the melanoma model, we showed better, for example, T-cell infiltration into the tumor. And so I think that's an excellent question. That's really our next step to prove with this syngeneic model. Is that what's happening?
Okay, yeah, that makes sense. I think another question came in the Q&A. So the question asked, is there a vision of this as just a treatment after diagnosis, or could this be used as a sustained post-treatment for someone who has undergone treatment and is trying to avoid relapse?
Yeah, the model that we're using right now is that it is sort of a maintenance treatment. So you've got that first line treatment, and then these vesicles, at least the empty vesicles, which we think is the easiest path through the FDA, is to use them as like a maintenance therapy. So I like you can tell from the bacteria stuff, right? I like something that's a little off the wall, something that's not everyone else is doing. And I also like to push for what I think is the most dramatic, the splashiest result. Jill has dialed me back and has convinced me, and I guess in our conversations with a number of people at the NIH and the FDA, is that it's not realistic for us to take, say, the bacterial vesicles and instantly get FDA approval. So I think our strategy is really, let's do the empty vesicles, which we think can be like a maintenance therapy to start, and hopefully will, in the case of osteosarcoma, suppress the metastasis to the lungs, for example. And once we have that through the process of approval, then we step it up, and then we do some of these modified vesicles that I think can be a primary treatment, and then the loaded ones, and then the bacterial ones. So Jill has like a 20-step plan for how this will progress. But I think initially that's our goal.
Yeah. Well, so since the idea is for it to be a maintenance treatment, I guess the thought is that there won't be many side effects to this. Like, I guess since you're flipping these macrophages or repolarizing them to be inflammatory, is there any worry about that? Like, I don't really know how that works.
Yeah, I think there has been some concern, not much less than you would with a traditional chemotherapeutic. We know from the animal studies, not only in the osteosarcoma model, but the ovarian cancer model too, is they tolerated the vesicles incredibly well, much more so than they did the cisplatin. There's a number of things that go on with, say, cisplatin treatment where you get peripheral nerve damage. So there has been some concern that the delivery of vesicles that may have a systemic immune response, right, where we're flipping macrophages or immune cells from one type of polarization to another. We haven't observed this, but we know that's a consideration. I think one thing that argues against that for us is that where something like cisplatin is a systemic treatment, right, it's going pretty much anywhere in the body, or most small molecule therapeutics act the same way. Our analysis when we're running these experiments is these vesicles are primarily trafficked to the tumor site. And because of that, we think that any of the peripheral nerve damage that might be a concern hopefully isn't going to be a concern, because we think we're acting almost exclusively within that tumor microenvironment.
Okay. Has anyone looked at instead of like cisplatin, like doxorubicin or methotrexate, putting that in the vesicle, and would there even be a reason to do that?
Yeah, well, we've looked at a number of different therapeutics. Cisplatin is kind of the one we've fallen back on. Doxorubicin is unfortunately very membrane permeable. So when they make these liposome treatments that have doxorubicin, I think primarily used for breast cancer, what they do is something called remote loading, where they crystallize the doxorubicin inside the liposome. So we've considered that with stuff like doxorubicin, and have looked for a collaborator that might have some experience in that. We have not been successful in finding someone that was interested in doing that. But there are other therapeutics that, of course, aren't as membrane permeable that we can try as well. So it's on the radar for sure.
Gotcha. Thank you. Christina, do you have any questions?
Yeah, those are great questions, Walker. I have a couple questions more just about how in creating the MEVs. So am I correct? I feel like I saw some of the cartoons where it showed it was leukapheresis, so you're actually extracting from the patient. So for the MEVs, is it important that they come from, are they patient specific, or can they be kind of universal off-the-shelf MEVs?
So our initial path was to be patient specific for a couple of reasons. One, We wanted to make sure they were compatible with the immune system of the patient. And there's a lack of, say, cell lines, right? We can't use THP1, which is the normal human macrophage-like line. So right now, our initial approach is going to be a apheresis from a patient, and then generating sorting sorting for monocytes, generating macrophages from that, and then generating vesicles and delivering back to the patients. And this is something else that I think is going to be beneficial for us with the ARPA-H award. Is part of that award is they're requiring us to reduce costs, because that's the biggest barrier. You can imagine apheresis, getting into a GMP facility, and the quickest way for us to do that is to generate an off-the-shelf macrophage that is also overexpressing one of our membrane markers, probably ICAM1, that we're interested in. So we are looking at right now a couple of methods of generating those. We have a new postdoc in the lab that their whole focus is generating a line that can be used, as you said, off the shelf, so we can avoid the whole idea of having to drag a patient in, do a blood draw, and then just have a limited window where we can treat. Where if we can do an off-the-shelf version, then we can generate unlimited number of vesicles, and it also probably allows us much wider treatment. You don't have to go to a special facility. You can get treatment at more of your local hospital. So I guess the long answer I gave to make it short is we're going down the one path initially, but as a side project, I think we're probably 6 months away from being able to generate something that's off the shelf.
Got it. Yeah, because I think the thing that's always interesting, I'm curious about with like all cellular therapies, is like the timing too of when you procure that, because it seems like it'd be kind of tricky because you know, if you're doing it, especially if you're looking in the clinical setting of doing this as maintenance after chemotherapy, because you know, as you mentioned, and the trickiness of delivering the cisplatin is that kind of kills the immune system and kind of kills the WBCs and macrophages. So like, are you kind of, you know, getting when do you know, when's the best time basically to kind of like harvest that stuff? Is it because post chemo seems like some of that's depleted?
Right.
On the other hand, beforehand, there is some potential like evolution that chemo might contribute to. So even like protein expression might change after. And so, you know, will the MEVs be as targeted to the right protein? So
yeah, I think that's an excellent point, which makes the off-the-shelf version even more important. The nice thing about the vesicles versus a cell-based therapy is the vesicles can be lyophilized. So we can basically freeze dry them and store them for months. We are in the process of figuring out how long we can freeze dry them. So we have an ongoing study where we're basically testing at various temperatures and for freeze-dried ones, and we're pulling them out and testing them to every 7 days, and we're trying to see where we start losing that efficacy in our cell culture studies for not being able to flip macrophage phenotype. So I think that'll give us a good idea of what the shelf life is for these once we freeze-dry them.
And I think one of the questions actually kind of got to this a little bit, but so different from, because you know we've had a lot of discussions before on RT and NK cells where the idea is that those potentially expand, and there's this like idea of like how much they expand and do they persist. And so for the MEVs, it sounds like it's just kind of is there any sense of like they don't expand, I'm guessing, and so is there any sense of persistence, or how often would you have to administer? And you talked a little bit about this, about being able to maybe like have fewer infusions, you know, if you were able to supercharge them with kind of better targeting. So what are kind of thoughts on, like, again, clinically, how this would be applied in terms of how many times you would have to infuse and over what, for how long could you do it for?
So our initial plan is once a week. We think that's a balance between having a patient come in for treatment and based on our studies in the animal models, what we think could be effective. So once we generate the vesicle, as long as it doesn't degrade, if we don't get any decomposition of the membrane proteins, then it's locked into what it was initially, right? There's no drift in the cellular phenotype like you would if you're expressing cells, or you have cells that you're trying to maintain in culture for long periods of time. I mean, they absolutely drift, right? We know no matter what type of cell you have, if you get something from ATCC and then you grow the cells for 20 passages, you get drift. Almost probably universally. With the vesicles, much the same as an exosome. Once the exosome is produced, once our vesicle is produced, you're not going to get any change in the properties of that vesicle unless you're getting degradation of the surface proteins. So our plan is to initially do the apheresis. That generates an incredible amount of vesicles as compared to what we can even from cell culture when we process those cells. And that where I don't know if we have dialed in exactly how many treatments one blood draw would give us, but that's ongoing. And then once we get to off the shelf, and we can basically make them and store them via lyophilization, and then hopefully there's no issues at all with degradation and no issues at all with them drifting since they're vesicles.
Fantastic. Well, we are out of time. So interesting. And I also just thought it was really cool how you shared your story about how this all came about. And you're knocking on Dr. Colasar's door, which turned out to be a great thing, because it sounds like there's just so many applications for these MEVs, both from being able to flip the macrophages and even just kind of more targeted delivery of some of these other cytotoxic agents. So thank you so much for joining us on Osteobites today and making it better for osteosarcoma patients. More information on this and all Osteobites can be found on our website at mibagents.org, on our MIB Agents YouTube channel, and at your favorite podcast place. Please join us on Osteobites next week for our last episode of 2024. We are going to be joined by Dr. Chelsea Burke from Memorial Sloan Kettering Cancer Center. She will be discussing her 2024 Outsmarting Osteosarcoma funded work on understanding tumor evolution in osteosarcoma. You can find our Osteobites lineup for the next few months on our website. And if you have any ideas for future topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Richards, for joining us today, and Walker as well. And thanks to all of you for taking time out of your day to join us. We hope to see you back here next week when we chat with Dr. Chelsea Burke. Thanks, everyone. Thanks, Dr. Richards. Thanks, Walker.
Thanks, Walker. Thanks, Christina.